Hello, everyone. I have the pleasure today of introducing our speaker this afternoon, Azhar Saeed Hammali, who is an assistant professor at the College of Science at the Imam Abdurrahman ibn Faysal University. Dr. Azhar obtained her PhD in mathematics in 2019 at the Oregon State University, USA. The research topics of Azhar are numerical analysis, parabolic equations, variational inequalities, and porous media. Today, she is going to present her recent results about numerical analysis and simulations of a model of biofilm growth in porous media. You are welcome, Azhar, and thank you for accepting our invitation. You have 50 minutes to present your work. Thank you very much.
Thank you so much. Thank you, Professor Nabil, for the invitation. It's my pleasure to present at the Al-Khwarizmi Applied Mathematics webinar. So today I'm going to present a joint work with Professor Marguzata Pishuniska from Oregon State University and Chao Chen from AB in Tissue Software. This talk is about numerical analysis and simulations of a model of biofilm growth in porous media. So first I'm gonna give you an overview of this talk. I'm gonna start by present or give you a brief introduction about what biofilm is, how it forms, its dynamic in porous media, and some of its applications. And then I'm going to present the model that describes the biofilm growth and the nutrient utilization in porous media. And you are going to see that it's a coupled system of two BDEs. One of them can be characterized as a parabolic variational inequality. So to analyze this model, I'm going to start by analyzing the BVI first, and then I'm going to generalize the results to the coupled system. At the end, I'm going to present some simulations. Now, this is the biofilm. As you see in this figure, it's a complex structure made of billions of microorganisms embedded in some slimy substance, extracellular polyramic substance, or EPS. Initially, when microbes attach to some wet solid surface existence of a nutrient, microbes consume the nutrient to grow, and then they produce this slimy substance, EPS, to protect them from the surrounding harsh environmental conditions such as dehydration, ultraviolet radiation, and also to boost their adherence to each other and to the solid surface. So once the EPS is produced, we can say that biofilm forms and it becomes mature. Biofilm keeps consuming a nutrient as long as it exists and then grow until it reaches a certain density that cannot be exceeded, because in porous media there is no enough room for it to grow. That means there is a volume constraint on the biofilm growth. Here is a cartoon of a porous medium where the black dot represents the rocks in this medium. The gray area represents the biofilm phase, and the white area represents the void space. Here where the fluid flows. Actually, it flows not only here, but also it can penetrate the biofilm phase to transport the nutrient to biofilm so it can consume it and continue growing. That means here we assumed that the biofilm is either permeable or partially permeable. Through experiments, it's been shown that when biofilm is close to its maximum, the majority of its growth occurs through the interface between the fluid and the biofilm. And when it reaches its maximum, it diffuses through this interface. So we can consider the problem that described this dynamic as a free boundary problem because the interface is moving over time. Okay. So now biofilm plays an important role in several applications in many fields. Here are some of them. Biofilm is used in the CO2 sequestration to plug the leakage in the CO2 subsurface storages. It's also used in this technique, microbial enhanced oil recovery, MOR. In this technique, water is flooded in the oil reservoirs to extract the residual oil. But what happens is that sometimes there are high permeable zones which prevent water from going to its right path. Rather than that, it develops water finger, we can say. So this prevents the water from extracting the residual oil. So to avoid this, biofilm is promoted in these reservoirs by injected bacteria and nutrient in these reservoirs. Another application, biofilm is used in the wastewater treatment through this technology, microbial-based green technology for wastewater treatment. Here, microbes are injected in the wastewater to convert the organic matter into a dense biomass, so it's easy, can easily removed and isolated through sedimentation. For more information about these applications, I invite you to read these papers and more. Okay, now this is the model that describes the biofilm growth and the nutrient utilization in porous media. As you see, it's a coupled system of two BDEs with advection, diffusion, and reaction terms. The first one in Nu1, the biomass concentration, and the second one in Nu2, the nutrient concentration. They are coupled here in the reaction terms, which are given in monod expression here. So this is to express the growth of biofilm, and this is to express the decay of a nutrient. Also, they are coupled here. The nutrient diffusivity depends on the biomass. Q bar here is the velocity of a fluid that can be obtained by the heterogeneous Brinkman flow model, which is this model. B is the pressure of the fluid, mu it's viscosity. QB here is the biofilm permeability. So as I mentioned earlier, the fluid can flow and penetrate the biofilm phase based on its permeability. Also, we have the volume constraint on the biofilm growth we donated by U star. And also we set this lower constraint since biomass cannot be less than 0. So that means we have this box constraint. This can be enforced by this red term. This is the subgradient of the indicator function defined like this. It's 0.
Azhar, please, the first equation, it's inequality, of course, here.
Yeah, this is inequality. Yeah, this is inequality.
Okay.
Yeah. Sometimes I call it BDE, but yeah, it's inequality. Yeah, thank you. This is the indicator function, okay, defined like this. Its subgradient, it's a multi-valued function, as you see here. So that's why we have this inclusion here. And this theoretically is equivalent to parabolic variational inequality, as we are going to see that later on. Okay, and also in a computational model, this can be replaced by a Lagrange multiplier and another problem called nonlinear complementarity problem that involves some semi-smooth function. Okay, now the biomass diffusivity plays an important role in modeling the growth and spreading through the interface. Here is the formula of the biomass diffusivity we consider here. It's a modification of this degenerate singularity, singular diffusivity obtained by or proposed by Albert Diemert in 2007. So it succeeds in modeling the spreading. As you see here, it blows up when the biomass is close to its maximum. But actually, using this type of diffusivity can be challenging because of the singularity we have. So it makes the analysis very hard, okay, since we need the diffusivity to be bounded from above and below. And also the degeneracy make it also very hard. And in practice, we need here to use very small such is not the case on most of so to overcome
इंस्पेक्शन करवानी थी तो उसका जो बटन है ना वो सादा लगा हुआ था
to overcome these challenges
उसका लगवाना
Please, the mic, please. Please.
भी काफी देर पहले लेके गए हैं तो वो पहले कहता बटन
Sorry.
yeah, to overcome this, we modify it by removing the singularity by introducing this parameter. It's a little bit greater than its maximum. So it can be the diffusivity remains constant when you reach its maximum, U1, I mean here. And also we remove the degeneracy by adding this small, very small constant. Alpha is a parameter. It's the strength of the spreading. And it's important to select alpha very accurate because as we see here, we conducted some experiments showing the effect of the values of alpha on the growth and the spreading. Here, this shows the thickness of biofilm over time with different values of alpha. When alpha, for example, equals to 2, as you see here, this is the solid line, we see that the growth started at this time, 0.6, and it diffuses slower than the other cases compared to alpha 3 and alpha 4. When alpha equals 4, we see that the growth delayed, okay, but it diffuses very fast. Now, so we need to select alpha such that none of these processes, the growth and spreading, overtake the others. So in this paper, we proposed a model called adaptive model that takes care of selecting alpha at each time step. So at each time step, we select alpha such that it's the minimizer of all alphas that solve the model, the system, such as this constraint hold. And we've seen that the adaptive alpha has to be greater than or equal to. Okay, in this presentation, we assumed that alpha is a constant, it's greater than or equal to. Okay. Now this model is challenging for several reasons. One of them is the volume constraint. As I mentioned, this can be characterized as a parabolic variational quality, and in literature, solutions to BVI's lack of regularity, in particular, the second derivative in time for the solution does not belong to L2. So this makes the analysis harder. And another issue is how to deal with the nonlinearity, okay, in the diffusivity, and how to deal with the coupling here, and also with the nutrient diffusivity. As I mentioned, in computational model, this can be replaced by some Lagrange multiplier and another non-linear complementarity problem that involves semi-smooth functions. So we need here a robust solver that can deal with all these things, the lack of smoothness, the non-linearity, and the coupling. In this work, we implemented the semi-smooth Newton method, and we've seen that it's robust. Another challenge is the analytical solution is not available. So this also makes computing the error harder, because we need to compare it with another numerical solution, compare the numerical solution with another solution that is computed at very fine grid. Last but not least, the complex geometries in the domains is another challenge. This makes the simulations much harder. Okay, now to analyze the problem, I'm going to start by analyzing the first BVI. Okay, we approximate the problem using mixed finite element method in space and backward Euler in time. We've chosen a mixed finite element method due to its conservative property, and also because we assumed that the system is isolated.
Azhar,
yes,
there is something that I don't understand. I don't understand what this sign mean, belong. What mean f of u belong to this quantity in the second equation?
Okay, yeah, because this is a multi-valued function. This red term is a multi-valued function, so it does not have only one value. Yeah, this is the problem here. So we don't write equal sign here because it's a multi-valued function. I don't know if it's clear now or not.
Okay, you can continue, but
yeah. Okay, so here we consider the Neumann boundary conditions. So mixed finite element method is the suitable method for this type of boundary conditions. So we rewrite the BVI in terms of U, and since we need to rewrite it in mixed formulation, we introduce this a new variable Q to be this total flux. So we get this system. We complete it by some homogeneous boundary condition and initial condition. And we set these assumptions. Q bar, the velocity belongs to L infinity. The source term here is a continuous function with respect to U, as you see here. M here is the monod function. U star, the maximum density is a constant. It's a positive constant. The initial function belongs to L2 and also satisfies the box constraint. And also we set these regularities on the solutions, U and Q. And the suitable functional spaces for the mixed finite element method are HDEF, defined like this, and L2. So HDEF for the flux and L2 for U. Since U has to satisfy the box constraint, we define this set K. Okay, so this convex, closed convex subset of L2, which is important in showing the well-bosedness of the problem. And since we have the homogeneous condition here, we defined H here, okay, X. So Q belongs to X and U belongs to K. And in the distributional sense, this subgradient of the indicator function is defined like this. So based on this one, this expression can be written equivalently into this parabolic variational inequality. Okay? And now by multiplying both sides of this BDE by a test function in X, integrating in by parts, we get that variational equality. And now we approximated in time using backward Euler, and in space used the mixed finite element method of the lowest order of Rabier-Thomas element due to the low regularity of the solution. So we discretized first the domain into rectangles in 2D or cubes in 3D. So this is RT0, the Rabier-Thomas space, where Q the numerical solution of the flux belongs to, okay, defined like this. And the finite dimensional space for the numerical solution for the scalar solution, M0, belongs to M0 here. It's the space of all piecewise constant functions. Okay, and actually here we want the numerical solution to satisfy the box constraint, so we defined KH. So this is the set that satisfies the constraint. It's the analogous one to K. Tau is the time step size, okay? And this is the discrete problem. Now we look for a solution QHN and UHN at each time step. This discrete problem is well posed. That means it has a unique solution for sufficiently small time step size tau. And we can show that by rewrite the system in this variational inequality that depends on this operator. This operator is defined like this. So it depends on the matrix form. Okay, A is the matrix corresponding the diffusion term, M is the matrix corresponding the monod expression. So now to show the well-posedness, we need to show that this operator is strongly monotone. That means it satisfies this inequality, and also it's Lipschitz continuous, okay, which is this one here. And since K or KH is a closed convex subset of L2, so it's easy based on this theorem from this book, it's easy to show that the problem has a unique solution. Now for the error estimate, as I mentioned earlier, we have some challenges here. The major one is how to deal with the nonlinearity of the diffusivity. We found that this paper linearized their unconstrained problem using Kirchhoff transformation. And we've also seen that this technique was suggested in this paper as well. Okay, so we follow this technique. Also, this paper suggested using a time integration to overcome the low regularity of the solution. So we use it as well. This paper also, since their problem and constraint and the diffusivity of the problem depends only on X and T, so to deal with this term that have this diffusivity, they also implemented the weighted L2 projection operator. Here, with the scalar BVI, we don't need to use it because by changing of variable, the diffusivity will become one constant. So we don't need, use it here, but we use it with the coupled system. So first, we define the Kirchhoff transformation like this. It takes you to this variable xi. We call it variable. It depends, we call it xi. So it depends on the diffusivity. Based on the properties of the diffusivity, K has an inverse. We denote it by B. And then we change the variable here. Now, the diffusivity of the new variable is now 1 constant. And this new variable satisfies this box constraint. So now we rewrite the system in terms of the new variable xi, and W is its flux, okay? And then we integrate the problem in time to overcome the low regularity. And then we approximate it, as I described before, backward Euler in time and mixed finite element of the lowest order of Rabier-Thomas element in space. Okay, so this is the discrete problem, the new discrete problem. Now we have some properties of the inverse of Kirchhoff transformation B, which are so important in our analysis. B belongs to C1 and non-decreasing function, Lipschitz continuous function, and satisfies these inequalities. And also we set these assumptions on the solutions xi and w, which are analogous to those of the previous variables u and q. And here is our theorem. The error in xi and w converges of first order. So this is first order because we need to take the square root of both sides. Okay, and to derive the error estimate, we used some auxiliary properties of some operators. We implemented 3 operators. The first one, the interpolation operator, and this one, H1. Okay, so it's defined like the normal component of any function in H1 coincides with the normal component of its interpolant on each edge. The second operator is the orthogonal L2 projection on M0. So this is M0, okay? MWH defined like this. These 2 operators are connected to each other by this property, minimal property, and we use it in our estimation. And also they both satisfy these estimations. Also, we used here the L2 projection in XH RT0, okay? And the outline of the proof is as follows. First, we go to the inequalities in both the continuous case and the discrete case. We replace the test function in the continuous case by the numerical solution of xi. The test function for the discrete case, we replace the test function by the orthogonal L2 projection of the solution xi. And then we combined these 2 inequalities. We use the definition of pi h, we get this inequality. And then we go to the equalities in both the continuous case and the discrete case. We subtract them from each other. We get this equality. Now xi h is the test function. Then we defined the W bar n is the average of the flux on the sub-interval, the time sub-interval from Tn minus 1 to Tn. IWN is the sum of those averages, okay? And sigma n is the error between this average and the numerical solution of the flux. Sigma hat n is the error of the integral of the flux from 0 to 2n. Okay, so it's the difference between IWN and its approximation. So now we replace this test function by the L2 projection of this error, okay, which can also be written in these 2 terms. We get this equality. We need now to estimate each term of these by the definition of pi and rho. We combined the result by the inequality obtained from the previous slide, and then we multiply it by the time step size tau and take the sum from 1 through n. We get this inequality. Now we need here to estimate each term here in the right-hand side and in the left-hand side. The left-hand side can be estimated using the properties of B, okay, and this lemma, so we get this inequality. Okay, so this is the error in xi. And this is the projection of the error in the flux, okay? So these two norms less than or equal some terms. So to estimate these terms, we use the auxiliary properties, okay, and Cauchy quartz inequality, epsilon inequality. By some appropriate choices of epsilons, we get this inequality, and now the proof is completed by using Grunwald's lemma. And now we move to the constraint nonlinear coupled system, which is this one, the one that I presented at the beginning. So to derive the error estimate for this system, we need first to linearize it in the same way we linearize the scalar BVI using Kirchhoff transformation. So we have this new variable xi, and W, it's a flux. We rename U2 to be a mu, and Z, it's a flux. Now we rewrite this system in terms of mixed formulation and integrate in time, we get this system here. So it involves this inequality. Now we approximated in the same way we approximate the BVI, okay? And also we use these assumptions on the diffusivities, biomass diffusivities, the nutrient diffusivities, and the reaction terms. So both the biomass diffusivity and nutrient diffusivity has to be bounded from above and below away from zero. The inverse of the nutrient diffusivity is continuous Lipschitz function, and the reaction terms are smooth functions on R2 and uniformly bounded. And also we make use of these assumptions for the solutions, which are these. I would like to mention here that some of these assumptions are set for the sake of analysis. We don't prove it theoretically, we just set them analogous to the solution of some unconstrained problem. But yeah, we need to prove it here. And this is our theorem. The errors in Xi, the biomass and nutrient, okay, and their fluxes converge of first order. And the proof of this theorem, here we use the same technique we used with the proof of the scalar BVI, but here we added this implementing the weighted L2O projection to be able to deal with the term that involves the diffusivity of a nutrient. And now it's time for the numerical experiment. Here we implemented the cell-centered finite difference method because it's easier to implement, and it's also equivalent to the lowest order of Rabier-Thomas element based on rectangles up to quadrature order based on this theorem. And also we implemented the operator splitting method. At each time step, we first solve the advection part explicitly. Once we get the solution, we plug it here in the reaction diffusion part. The reaction diffusion part can be written in this computational sense by lambda, in terms of lambda, as I mentioned earlier. And also it involves this equation that involves this semi-smooth function. Okay, now we need to solve this system in terms of U1, the biomass, nutrient, and lambda at each time. Okay, at each time step. And to solve this system, we implemented the semi-smooth Newton method. And here is a 1D simulation. Here we assume that we have a tube of length 1. Its boundaries at x equals 0 and x equals to 1. We assume that the medium is rich of nutrient, and the flux is constant. The flux rate is the flow rate is constant. Okay. And we assumed that at initial time, the biomass is located in this region only at its density is 0.8, where the maximum density is 1. The magenta line here represents the threshold of maturity, okay, which is 0.9. Okay, and we see that over time, I forgot to mention that a nutrient is injected constantly in this wall. That means the medium is rich of a nutrient. Okay, so over time we see that the biomass grows upward by consuming the nutrient, and it becomes mature at this time. It continues growing, okay, until it hits its maximum density, which is 1. So once it hits its maximum Lagrange multiplier lambda, which is in red here, in blue, it changes from top growing upward, rather than that it diffuses forward. And also we compute the errors for the biomass and the nutrient in L2 norm. Here was the various with different values of H, the spatial step size, and tau, the time step size. We've seen that the order of these two errors is 1. Okay, or more than 1 sometimes, as we can see here. We also computed the errors in respect of L-max, and we see that L-max norm, and we see that it's roughly 1, but the error in L2 norm exceeds that of in L-max norm. And now here is a 2D simulation. In this simulation, we coupled the Brinkman flow model with the biofilm nutrient model, which is this one, and we implement the splitting operator. At each time step, we computed the velocity from the Brinkman flow model first, and then we plug it here in the advection part, and then we plugged the solution from this part to the reaction diffusion part. And we see that in this simulation, we have a medium of single pore. We assume that at initial time, there is no any nutrient, and the biomass fill 10% of the medium, and its density 0.6. And the nutrient is injected over time, constantly at this wall. The fluid flows from left to right, and this is the initial rate. We see that over time, after 4 hours, biofilm forms, and it accumulates at this region near the solid surface. Okay, this is only to show the domain of a biofilm and the non-RAC solution, the non-RAC domain and the RAC domain. And in this simulation, we used the same data we used with the previous one, with the previous simulation, but here we test the effect of the permeability and the growth of biofilm, and also in the flow of the fluid. So we consider 3 cases: the permeable case, the partially permeable case, and the impermeable case. We've seen that the biofilm forms and glut completely the solid surface at this time. So it's completed the glucking at this time, 9.1, which is faster than the other cases, the partial permeable case and the impermeable case. And this is what we expect. We expect that since it's permeable, it allows the fluid to flow in it to transport the nutrients. So biofilm can consume the nutrient and grow faster than the other cases. Also, we see the effects of the permeability on the flow. As you see here, the flow stopped completely in this case, the impermeable case. Okay. To summarize, in this presentation, we present the biofilm nutrient model. We approximated using mixed finite element method with the lowest order of Ravier-Thomas element. We derived an optimal error estimate, and we illustrate the result with a numerical experiment using realistic data. Our current work is to numerically analyze the coupled Brinkman flow model with the biofilm nutrient model. And also, I'm working now with my team in the department in using different solver than semi-smooth Newton method. Since semi-smooth Newton method is locally convergent, so we need to look for other solvers. Also, we want to relax some assumptions on the regularity of the solution required for the convergence. As I mentioned, some of these the assumptions are set for the sake of the analysis. And future work is to numerically analyze the adaptive model that takes care of selecting alpha, the strength of the spreading. And thank you, and I'm open to any question.
Thank you, Azhar, for this beautiful talk and for these very beautiful results too. Is there some questions, please?
Yes, Nabil, I have a general question, please.
Okay.
What do you mean in your future work by adaptive model? How you can use this technique of adaptive model?
Adaptive, yeah. Yeah, I mean, the adaptive model, okay, yeah, I can go to that slide that shows the adaptive model. Yeah, here. So the adaptive model that solved the system not only for B, the biofilm or the nutrient, but also solved the system for alpha as well. Okay, since alpha is unknown, that means alpha is unknown. We need to choose alpha accurately such that this constraint is held without actually needs to the subgradient of the indicator function. Because in this paper here, Albert Demart, this diffusivity does not need to have the Lagrange multiplier or the subgradient of the indicator function. This models also the constraint. Okay, but as I mentioned that we have some challenges in dealing with the singularity and the degeneracy, so we adapt or modified this diffusivity. But here we need to choose alpha without needing to the Lagrange multiplier. Okay, and how to choose this alpha by the adaptive model.
Okay, thank you.
Cyril Bandelier have a question. Can you ask your question, Cyril?
Yes. So thanks for the talk. I wrote it, but I can read what I wrote. So can you say a word on the images that you showed at the end of your talk? They are simulated via which software? And what is the mesh? What is the mesh for it and the time that you need to generate them?
So we used MATLAB. And what's the second part of your question?
So what is the mesh, the resolution of not the image, but the mathematical resolution of your lattice, if you want?
The mesh, yeah, very fine grid, but yeah, I don't remember exactly. Yeah, it's very fine. Okay. Yeah. It's very fine grid.
Yes,
Okay.
that's
But
the
Cyril,
because
she
there
said
is
at the
a
beginning that the
first
mesh is just
for it.
square,
Yeah.
finite.
Yeah, here, yeah. So here we discretized the domain using rectangles or in 2D. So this is a 2D simulation. And
Okay.
here we implemented cell-centered finite difference method, not the mixed finite element method, because as I mentioned, it's easier to implement and it's equivalent to RT0, the mixed finite element method, up to a quadrature order.
So so what is the time that MATLAB needs to do this? For example, the first image. So there are several parameters which can play a role. So
Yeah, the
you have
time.
some PDEs. So how many digits of the solutions are correct, and what is the size of the grid? They are the two parameters which could deeply impact the time that you need to generate the image. So what is a little bit the situation for those ones?
Okay, so the time that we need to run the MATLAB here, actually it's very fast. Yeah, it's in minutes, and you can find the you can find the code in GitHub. It's developed by my collaborator Chawachen. Yeah. And yeah, I don't remember exactly the size of the meshes and that time step size, but yeah, but I remember that it's very robust, it's very fast.
Okay, thank you.
Hello, please. I have a question.
Yes.
My name is Mohamed Ngom. I'm from Senegal, University Alioune Diop, Bambey. My question is about slide 6. Just a precision.
Yeah, okay. Slide 6.
Slide 6, yes. Yes, here. What about this zero when we have U star equal U1?
U star. Okay, where U star
Equal U1. About this zero.
Oh, okay.
Yes, here.
U star. Okay. A plus, so U star is the maximum density. In our simulation, we used U star to be 1. Okay, but does not mean to have, it's not necessary to be 1.
In the middle term, you make U1 between
He's
0
worrying
and...
about the fact that you have a division by 0 in your formula.
Okay.
Okay. So say that again. Or what's your question again?
I think that he is
If U star equal U1,
you could divide by 0.
the expression is fine.
Mm-hmm.
There is singularity.
Yes.
Yeah. So if U1 equals to U1, you mean?
You start.
U star. Okay, so the definition is here. Okay, U star bar is greater than U star. So we don't have the...
Okay, it's U star, but okay.
On the previous, on the other side.
But you don't define U star bar. Okay.
Yeah, U star is a parameter a little bit, yeah, here.
Okay.
Greater than U star.
Okay, it's not the same problem.
Thank you. I'm okay.
And on the other side, when you mentioned this Eberl-Desmarais result, there is a bar or not?
Where?
Because here we see the singularity possibly.
Same
The
slide.
same,
on the same
the
slide,
same slide.
the previous slide. Yes. So here you had for the Eberl-Desmarais result, it was U star minus U1, and then we see that indeed there is a polar singularity.
Here?
No.
No,
The left.
Yeah.
the left.
Eberl-Desmarais
Yeah,
result.
this one is singular here.
Yeah,
This one
exactly.
is singular. Yeah. So that's why we modify it to remove the singularity.
Yeah.
Yeah, so it's singular here. Yeah, we can see that in from the figure as well. So this is proposed by Albert Diemer. And so we take it, but we modify it to remove the singularity.
Yeah, exactly.
So you exclude the region very close to U star. Okay?
Yeah.
Can I ask a question, Azhar?
Sure.
You presented first a discretized model using a mixed finite element method.
Yes.
Exact?
Yeah.
But for your numerical result, I understand that you use finite difference.
Yeah, self-centered.
You said because it's easier to
implement. Yeah.
implement. But do you obtain the because I missed some slides. I missed some slides. Do you obtain the convergence results for your discretized problem for the mixed finite element problem?
Yeah. So we use the self-centered finite difference method because it's equivalent to Ravier-Thomas element. Okay, so it's equivalent based on some paper by Wheeler. Okay, so we use it, it's easier to implement. In my PhD thesis, I used the mixed finite element method, not self-centered finite difference method, but in the simplest case. Not with the advection case and where the diffusivity is non-linear. So the diffusivity there was just constant, and we don't have advection term. So yeah, I use it in my PhD thesis, and we obtained actually a convergence result.
Okay. Is there other questions? If no more questions, we can stop perhaps here our webinar today, and we hope we can see you next week.